I just want to look briefly at the major histocompatibility complex, particularly the class 1 and 2 molecules. So if we look at the basic function of both molecules, it's obvious that both molecules are designed to present antigenic peptides to T cells. This may lead to an immune response that may result in the activation of certain immune cells and the production of antibodies. It's important at this stage to note that peptides associated with the class 1 MHC molecule are products of the endogenous antigen processing pathway, and peptides associated with the class 2 MHC molecule are products of the exogenous antigen processing pathway. These pathways will be covered in another video. As you can see in this diagram, both CD4 and CD8 molecules are present. These are T cell co-receptors that act as docking sites during the MHC to T cell receptor interaction and help to stabilize this interaction. If we just turn our attention to the diagram on the right, we can structurally compare both of the MHC molecules. The class 1 molecule contains a 45 kilodalton alpha chain and a 12 kilodalton beta domain, whereas the class 2 molecule has a shorter 33 kilodalton alpha chain and a 28 kilodalton beta chain. Both molecules have a transmembrane domain that has approximately 25 hydrophobic amino acids. This domain acts as an anchor to the molecule, giving the molecule stability when interacting with T cells.
Now, one area in which both molecules vary greatly is in their association with the peptides they present. If this diagram represents the peptide binding cleft, we can look at both molecules' methods of peptide presentation. The MHC1 molecule is only capable of presenting peptides of 8 to 10 amino acids. In comparison, the MHC2 is capable of presenting peptides of 13 to 18 amino acids in length. Furthermore, the MHC2 requires specific amino acids at either ends of the peptide binding cleft to act as anchors to secure the peptide in the cleft. These are usually hydrophobic residues such as leucine and isoleucine, whereas the MHC2 molecule has no such requirement. Finally, as I've tried to demonstrate in the drawing, both the MHC1 and MHC2 present the peptide in different conformations. The MHC1 presents the peptide in such a way that it anchors itself almost protruding away from the floor of the cleft, which allows peptides that are slightly shorter or longer to be accommodated for, whereas the MHC2 molecule has a binding cleft that is open at both ends, allowing peptides to extend beyond the ends of the cleft. Like described in one book as similar to a hot dog in a bun.